So this could be one of the easiest or one of the most confusing lectures throughout AMP1. And the reason why it can kind of go either way, it just depends on, are you only going to follow what you are required to know for the class, or are you going to try to make this a lot more difficult on yourself? Because spinal nerves, cranial nerves, and the brain, these are all very, very, very complicated structures. But the ones you're going to be covering in AMP1 lab and AMP1 lecture are really not that bad, because we don't know a lot about the brain right now. Even though we're doing a lot of research into it, there is still a lot we're not sure about what's going on. There are still cell types that haven't been discovered yet in the brain, so there's a lot of mystery about how does we interpret consciousness. How do we have personality? Do we have a spirit? All of these things, these metaphysical things that have been talked about in philosophy for centuries and decades. These are things that we're not going to cover in this class. All we're going to do is, what are the lobes? So if you do just listen to this lecture and say, what are the main things that I need to take out of it? This will be something that there's 4 things to know here, there's 5 things to know here, and there's 8 things to know on this model. And if you do it that way, this becomes a much more enjoyable process. Let's get started. So I don't have this model for the meninges, but the meninges are very, very, very important. So knowing what are the meninges on top, middle, and on the surface of the spinal cord and brain, it's extremely important. And it goes from dura mater, arachnoid matter, pia mater. And if you speak German, dura mater sounds kind of familiar because dura is durable and matter is mom, so it's a tough mom. So pretty much you have a very tough membrane that covers the brain and spinal cord. And a lot of the times if you're doing a epidural or you're doing some type of spinal tap, you have to push the needle through that dura mater. And a lot of the times when you're doing that type of procedure, a lot of the doctors and nurses, they're listening for a pop, and it's getting through that dura mater. That's why a lot of the times if you're taking an in-person lab, doing the sheep dissection, brain dissection is important because you get to cut through these different meninges, and a lot of the times with the scalpel you're like, it's not sharp. It's very sharp, but the dura mater is very, very, very tough to get through. The arachnoid matter, on the other hand, when you pull back the dura mater, the arachnoid matter sits and gets stuck to it. So when you pull it back, there are these little fibers that are holding on to the pia mater, which almost looks, when you're peeling up, there's little spider webs attached to the pia mater. And that's why the arachnoid matter kind of looks like little spider webs or arachnids or spiders. Pia matter is going to be surrounding the brain. So if you do a sheep dissection and you look at the brain and you're like, just like looking at it kind of like if this was a human brain, you're like, I don't see anything. And what you would do is you would take your probe, kind of like this pencil, go into one of those little grooves called the gyri, and then lift up, and you'd see that there's this little translucent membrane that's on top of the brain and spinal cord. That's going to be the pia mater. Let's take a look at it. So here's your spinal cord. This is going to be a cervical vertebrae because it's 1, 2, 3 holes. You have the dura mater, which is going to be on top. Arachnoid matter, remember, is going to be stuck to the dura mater. There's going to be little fibers connecting the arachnoid matter to the pia mater, and the pia mater is going to be surrounding the spinal cord and also the brain. There's also going to be a space between the arachnoid matter and pia mater where you're going to be able to have cerebral spinal fluid go around the entire structure. This model is going to be the one that you're going to be asked on the test. So knowing all these different things, it's very, very, very important. And if you kind of look behind me, I have all of these different models that just show you the spinal cord. And pretty much, if you look here and you're like, I see a butterfly, I see some white things, and I see some bulging back here, that's all you need to know for this topic. So you start here with the dorsal root ganglia. And a ganglia is just going to be a group of cell bodies which makes the neuron bulge. So a dorsal root ganglia just has a lot of cell bodies which makes it that bulgy shape, which means that every time you see the spinal cord, you see bulges, it's always going to be going to the back. That's how you can always tell the back from the front. And once you do that, every other structure is just memorization. Posterior median sulcus, anterior median fissure. Yes, you might study enough to be like, what's a sulcus, what's a fissure? Sulcus, just going to be an indentation. Fissure is going to be a deeper indentation where you can almost put your entire fingernail in. This sulcus in the back, very, very, very unimpressive. It's just kind of like a little bump like that. Sulcus, on the other hand, is going to be more of an indentation. But as long as you know back, because of the dorsal root ganglia, posterior median sulcus, anterior median fissure. Then you need to determine what's a horn, what's a column. So a horn is going to be the dark matter. Columns are going to be the light matter. And if you remember light versus dark for the brain in neurons, anytime there's myelin, it's going to be a lighter color because myelin is going to be more heavier in lipid. So it's going to be for the axons having myelin. And if you remember back even more, the myelin is going to be for traveling signals. So they're either going to be going up the spinal cord or down the spinal cord. And this is going to be dark matter because this is going to be more cell bodies, which is going to be doing more processes or connections to outside the central nervous system. And the names are pretty easy. Dorsal column, lateral column, ventral column. The dark is dorsal horn, lateral horn, ventral horn. And if you're like what it says, dorsal, lateral, and ventral horn over here, just so you don't get tricked up. There's a dorsal horn there, there's a dorsal horn there. Even though it's only a dorsal column on this side, there's still a dorsal column on the other side. So if you're looking at the model, I can say, name this, that's a dorsal column. Name that, that is also a dorsal column. So once you kind of memorize that, doesn't really matter what model I grab. All of them are going to be exactly the same. I should probably hold it like this because you can see dorsal root ganglia bringing it to the back and then the front. So sometimes even though I'm doing it through a camera, I can get mixed up. But you always want to start with the dorsal root ganglia. So let's take a look at some of the models. And these are the models, two of the models that I showed you, where you start with the dorsal root ganglia, posterior median sulcus, anterior median fissure, and then determine, is it a dorsal column or horn, a lateral column or horn, or a ventral column of horn, and then you're done. The other way I can ask the spinal cord is the end of the spinal cord. Your end of the spinal cord ends after T12 and L1. And the reason why is because when you're a child and you're 9, 10, you're a certain height. But then after that, you keep growing up. And when you're growing up, your spinal cord's not continually being remade and lengthened. The only thing that happens, the axons get longer and longer. So that is why your spinal cord ends at T12 and L1. And because it ends, it's going to make a cone shape around T12 L1. That cone shape is called your medullary cone. All the little fibers that hang down is now going to be referred to as your cauda equina, or horse's tail. And then if you looked at the top or the tip of that cone, there's going to be one nerve that goes all the way down the middle, and that is going to be the filum terminale. And the model we have for that is this guy. And this guy is kind of interesting to look at. So here you can see that the spinal cord is ending and it's making a cone shape right there, or your medullary cone. After that, you're going to have cauda equina all going down. But if you look down the center of it, I'll kind of go closer, you will be able to make out there is one nerve coming out the tip of the conus medullaris, or medullary cone, and that is called your filum terminale. So expect those different ways for me to ask spinal cord questions. That model can also be used for the plexus, like cervical plexus, brachial plexus in the shoulder. You also have lumbar and sacral plexus in your lower back, and then your hip. I know for the lecture class, I usually talk about certain ones like phrenic nerve for breathing and the sciatic nerve for your lower back, because the sciatic nerve biggest nerve in the body, also going to be the cause of the majority of lower back injuries, because back here, if you remember back when we were doing the os coccyx or the innominate bone or the hip, there's a greater sciatic notch and lesser sciatic notch. So the sciatic nerve loops around a bunch of muscles, can be pinched there, and can cause a lot of issues. Let's get to the brain. So the brain. We have many different models that can kind of go over this. And what you're looking at is lobes. Another thing what you're looking at bumps and grooves. So the bumps are going to be the gyri, the grooves are going to be the sulci. Kind of like how the back here was the posterior median sulcus. Sulcus just means shallow groove. So every time you see a shallow groove where you put your finger in, all of those are going to be sulci. The bumps on top will be gyri. It doesn't matter what brain I have, you'll still be able to make out the bulges are going to be the gyri, sulci will be the individual grooves. Another thing to kind of look at are the lobes and then some of the other things. So if you look at this one right here, you can see there is a central sulcus between the dark red and the dark blue that runs all the way down. You also have, if you look down, right there, going all the way down between central sulcus. Then you're also going to have the cerebral hemispheres right there. And then another thing you can see on these, you can see the individual lobes, where you see the frontal lobe is going to be red, parietal lobe will be blue, occipital lobe will be yellow, temporal lobe will be yellow, occipital lobes will be tan. You should know those lobes no matter how I look at it. Corpus callosum is going to be on the next slide. Corpus callosum is going to be on the inside right there. And it's going to be this little white track right there. And over here, this is the corpus callosum. Corpus callosum, if you look, is like a little ear. So if I held this up, you would see corpus callosum right there. You would then see back here is a pineal gland, just like there is right there. You'll also be able to see down here is going to be the hypothalamus. Go up and behind that little dot, that's going to be the thalamus. Below the hypothalamus is going to be the optic chiasm, which if you looked, optic chiasm is right there. Behind the optic chiasm is the mammillary body. You do have this little area right here. This is called the cerebral aqueduct, and the fourth ventricle is going to be right in front of this little network of white lines. Those white lines are going to be the arbor vitae, and the arbor vitae is going to be within the cerebellum. You have the midbrain, pons, and then the medulla oblongata. Now, if you looked at those and you're like, okay, frontal, parietal, occipital, temporal lobes, I got every other thing here. Pituitary gland is not going to be on any of the models. You know, I can hold that back up. There's no pituitary gland there, and it's because they fall off. But one thing when you're studying this, I showed up at the beginning that there are two sides. If I break them apart, what if I ask this one on the test? Would you be able to see the mirror image of this? Would you be able to say that this is frontal lobe, parietal lobe, occipital lobe, temporal? There's your corpus callosum, but instead of the pineal gland being in the back, it's now going to be in this view in the front, pineal gland. If I go closer, you can go thalamus, hypothalamus, optic chiasm, mammillary body, midbrain, pons, medulla oblongata. Cerebral aqueduct is right there. Fourth ventricle, arbor vitae is the white line, cerebellum is in the back. So I do have a lot of pictures, I do have a lot of ways to look at it that kind of show you that don't just memorize one picture, because no matter which model I give you, they have two sides. So a lot of people get very confused and get very kind of angry when they're studying this and be like, this isn't the one I studied. And it's because it's the mirror image. So when you're studying this, it's good to hold both of them and then see if you can kind of mentally look at how they're mirror images of each other. Like I know your hands are mirror images of each other, but when you look at them, they're not that confusing. But when you look at the sagittal section of the brain, it can be confusing. And this is the picture, that's just the opposite one. So corpus callosum, pineal gland, thalamus, hypothalamus, optic chiasm, mammillary body, midbrain, pons, medulla oblongata. Cerebral aqueduct is right there. That's going to be the fourth ventricle, and those white lines are the cerebral aqueduct, and the arbor vitae is the white lines, and then the cerebellum is that little thing on the back of your brain. You do have these Roman numerals, and these Roman numerals are going to be at the end of this lecture, and these are going to be the cranial nerves. The cranial nerves are very, very, very difficult to find, but there is a very easy way to see them. And you can see 1, 2, 3, 4, 5, 6, 7, 8, there's 9, there's 10, there's 11, there's 12. And if you're like, what? I did that because I know what the Roman numerals are, but we'll talk about that in a second. Here you can see frontal, parietal, occipital, temporal, and cerebellum. You see the central sulcus. You see the sulci, which are these grooves. The gyri are the bumps. Here's a human brain, just to kind of show you what it would look like if we were looking at a human brain, a real human brain. And then here is something that's kind of confusing. This is going to be, how does the cerebral spinal fluid circulate around your brain? And you heard me here, and you heard me say the word fourth ventricle and cerebral aqueduct. How do we get 4 ventricles? Well, first, you have this structure right here. And if you look, it kind of looks like ram's heads, or ram's horns, where they kind of go up. And these are your lateral ventricles. And if you think, how many lateral ventricles do you have? 2. So that's 1 and 2. Your third ventricle is when they come together. So right in the middle will be your third ventricle. The third ventricle is then going to send stuff into the cerebral aqueduct. The cerebral aqueduct ends at the fourth ventricle. Fourth ventricle is right in front of your cerebellum, and then it eventually goes down to the spinal cord. Here you can see it again. So if you're just looking at this model right here, you have your lateral ventricle, third ventricle, cerebral aqueduct. If you look up there, fourth ventricle, and then it goes down. So you should still know this model too. And it's going to be only lateral ventricles, third ventricle, cerebral aqueduct, fourth ventricle, and that's it. And I told you cranial nerves. There's 12. And now, if you look, there are 2 sides of the brain. One side of the brain is going to have Roman numerals on it, or 1 all the way down to 12. The other side is not. And if you look at this, 1, 2, 3, 4, 5. If you get the side that has Roman numerals, all you're going to be doing is labeling. And as long as you understand Roman numerals, you'll be able to come up with olfactory, optic, stuff like that. Your lecture class might make you, are they sensory, are they motor, or do they do both? Not in the lab. All we're going to do, can you label them? So first, it starts with cranial nerve number 1, olfactory. Optic nerve is number 2. Ocular motor is number 3. Number 4 is trochlear. 5 is trigeminal. 6 is abducens. 7 is facial. 8 is vestibular cochlear. 9 is glossopharyngeal. 10, X marks the spot, vagus or wanderer. And in your lecture class, you're going to learn the wanderer or the vagus nerve wanders down into your neck, your throat, your heart, your digestive tract. So there's a lot of things. If you ever have a vagal vagus reflex, it makes you pass out and you feel something in your stomach, your heart starts to race, your throat gets a little dry, and then eventually it kind of resets your brain where there's too much information going in. Number 11 is spinal accessory or accessory nerve, and then number 12 is hypoglossal. And the way you do it, 1 tick mark is 1. A V is 5. An X is 10. If you have a 1 in front of a V, it's V minus 1. That's why I said trochlear nerve is number 4. I knew abducens was number 6 because it's 5 plus 1 abducens. And then X is 10. If you put the tick mark before the X to the left, it's 9 for glossopharyngeal. Number 11 is going to be spinal accessory. So I kind of mentioned some of these are going to have Roman numerals. So if you look, you can see the Roman numerals on that model. If you take a look at this one, you can see the Roman numerals on that model. On your midterm, on your final, I don't know which one I'm going to have on your test. If I have the one with the Roman numerals, I'm expecting you to memorize 1 through 12 with those Roman numerals, what they mean. So if I say, let's just say this one right here is VI. VI, you know, is 6. You put abducens. Now, what if I have this one? How do you memorize the location of cranial nerves? Well, it's actually very easy when you go only to 5. Olfactory, optic, this is the pons right here, and medial to lateral is 3, 4, 5. So if you looked on this model, brain, 1, 2, 3, 4, 5. See, that's a white line right there, so 3, 4, and 5. And when you look at it like that, that's not that bad. That's not that difficult for you to memorize all the cranial nerves. Here's another view of them where you can kind of take a look and say olfactory, optic, ocular motor, trochlear, trigeminal, then abducens, facial, vestibular cochlear is 8, glossopharyngeal is 9, vagus is 10, spinal accessory or accessory is 11, hypoglossal is going to be number 12. Just to review the neuron, you have the dendrites bringing information into the soma. You have the axon hillox that kind of adds all of that signal together, and then it's going to be an all or none response that signals down the axon. The axon for fast movement is going to use myelin, and the myelin allows the signal to jump the nodes and goes through the node of Ranvier. And then at the end of the axon, there are synaptic knobs that have synaptic vesicles that allow the neuron to talk to one cell from its cell to another cell. And the Schwann cell is what is going to maintain the myelin sheath on every peripheral nervous neuron. And there's just a same view of the synaptic knobs. So that concludes the brain and spinal cord. And if you remember, don't go into any more detail. Memorize exactly what I kind of went over, and then you should be okay, and you should be have a good chance to do well on this test instead of making it, oh my God, neuroscience, it's so difficult. It's really not. Take it little by little, and good luck.